Hello everyone, welcome back to the channel and to the fourth episode in my ongoing series, The Causes of Evolution. If you're new here, my name's Zach Hancock. I'm a postdoctoral researcher at the University of Michigan and an evolutionary biologist. And on this channel, the sorts of things you'll find is a bit of evolution education, some pseudoscience debunking, and the history and philosophy of science. Now, I want to begin this episode a little bit differently than how I've done with the previous in the Causes of Evolution series, with a case study. And I think that this introducing this topic in this way will sort of prime us and act as a good segue into the rest of the discussion that we're going to have in this presentation on the mutational mechanism. So all of us should be familiar with the little fruit fly Drosophila melanogaster. It's a model system in biology that we've been studying since the late 1800s. We know effectively everything there is to know about its genomic structure, its development, etc. And this makes it an ideal system for studying how new genes can emerge via the mutational process. Now, there's about a dozen other species in the Drosophila genus, and they all have a certain arrangement of two key genes, and those are pictured up here on the top left, the CDIC and the annex gene. The first one is the cytoplasmic dynein interchain complex, and the second one is the cell adhesion protein annexin X. Those are mouthfuls. Just know that they're involved in microtubule structure formation, and they have lots of additional functions as well. But these two genes are in tandem. So you have the first one, then you have some intergenic region, and the annex gene is following right after it. Now, on the top, you can see the little boxes represent exonic regions, so these are the actual coding regions, 1 through 7, and then you have V1 through 3. These represent alternative splice sites. So all the Drosophila species have this particular genic arrangement, except for Drosophila melanogaster. You see, melanogaster has an additional gene sandwiched between those two. And what Nerminsky et al. In 1998 found was that this gene appears to be constructed from rearrangements of the original two genes that exist in all of the Drosophila genus. So I want to read you just a little bit from their abstract, in which they define what this gene is and how they came about finding it. So they note that they identify a brand new gene in Drosophila melanogaster that codes for a sperm-specific axonemal dynein subunit. The gene has a new testis-specific promoter derived from a protein coding region in a gene encoding the cell adhesion protein annexin X, which is that annex gene from the original two, and it contains a new protein coding exon derived from an intron in a gene encoding a cytoplasmic dynein intermediate chain, which is the CDIC. The new transcription unit, designated SDIC for sperm-specific dynein intermediate chain, has been duplicated about 10-fold in tandem array. And they end with the discovery of this new gene supports other results that point to the rapid molecular evolution of male reproductive functions. So they propose that this new gene originates as follows. So initially, you have a tandem duplication of these two regions. So you can see you have the original CDIC and the annex, followed by an additional CDIC and annex. And then there are 3 unique deletion events that cut out chunks of these two and sandwich them together. As I just read from the abstract, they acquired both a new exonic region that was previously an intron in the CDIC region, as well as a novel promoter from the annex region. These two things together, just sandwiching these proteins, followed by 10 additional substitutions, all of these mutational processes has generated a novel dynein subunit that is important in spermatogenesis in Drosophila melanogaster. So this is a bunch of independent mutations, including duplication, substitutions, deletions, as well as a retrotranspositional event, all of which have generated a novel protein. The structure of that protein is shown down here on the bottom. You can see that it is just novel arrangements of the original exonic sequence, as well as the acquisition of an intronic sequence into a new exon. Collectively, what this case study shows us is that multiple independent mutations are capable of generating completely novel proteins that are important and necessary for proper functioning of the organism. Furthermore, this entire new gene must have originated since the common ancestor of Drosophila melanogaster and all the other Drosophila species, since this gene is unique to this particular lineage. So I just wanted to begin this conversation with this really cool case study. We're going to look at several other ones further on because fundamentally mutation is the process of evolution that generates novelty, and we need to demonstrate that it can, in fact, generate this novelty. So with that being said, let's now take a step back and refresh ourselves on the normal structure of these presentations. So as with the previous presentations, the assumptions of Hardy-Weinberg are central. From these assumptions, the violations of these assumptions are what tell us what are the mechanisms of evolution itself. So we found previously that when gametes are not paired at random, the violation of the first assumption, that is non-random mating, and that it is a weak though important mechanism in evolution. Secondly, we found that the violation of the assumption that gametes aren't preferentially chosen from the gene pool is natural selection. Today, we're going to focus on the third and fourth assumptions, the third one being that allele frequencies are equal between the sexes, and the fourth one that alleles don't change from the individual to the gene pool. So before we talk about the main focus of this video, which is the mutational mechanism, let's start with this third assumption that allele frequencies are equal between the sexes, and let's see if this also is a mechanism of evolution like the violations of the previous two assumptions. So what happens if the allele frequencies are different between the sexes? So I've shown sperm and egg over here on the right. You can see that since those P and Q frequencies are different from one another, we have to represent them differently. So the contribution of P from the male is P sub M, from the female is P sub F. We do the same things with the Qs. So in the case of random mating, we can just pair all the possible combinations, and what we get is for the homozygote, we'll just denote that as capital P is equal to P sub F, P sub M. Now remember, if they are equal in frequency, this is just P squared, right? But since they are not equal in frequency, we have to write them as separate. For the heterozygote, you have a capital H, which is equal to P sub F, Q sub M, plus P sub M, Q sub F. Remember again that equal allele frequencies would be 2PQ, and then capital Q is equal to Q sub F, Q sub M. So in expectation, we should be able to take the average of P sub M and P sub F to get the expectation of P bar, which is the average frequency between the two. So if we want to know how this unequal frequencies between the sexes is going to impact our expectation under Hardy-Weinberg, we can then look at the difference between the observed, which is capital P, and what we expect to see, which is P bar. So we just write that out as capital P minus P bar, which is equal to P sub F, P sub M, minus P sub F plus P sub M divided by 2, which is what this P bar here is. We then square that because this is the frequency of the homozygotes, and that's what we would expect to see under Hardy-Weinberg equilibrium. We just rewrite this a little bit as well as distribute out our terms. And then let's plug in some numbers and see how it affects it. So let's assume that P sub M is 0.7, P sub F is 0.6. If that's the case, we plug in our numbers and we find that the difference between the observed and the expected is negative 0.0025. That is to say that when allele frequencies differ between the sexes, this causes a depletion of homozygotes relative to what we expect under Hardy-Weinberg. So fewer homozygotes. So what about in the heterozygote case? So we do the exact same thing. Since it should be 2PQ, we just take the expected P bar and Q bar. We do a little bit of rearranging and we get P sub F plus P sub M multiplied by Q sub F plus Q sub M divided by 2. So again, what's the difference between the observed and the expected? We just write that right down here. So remember, this is the observed from here. This is the expected, what we just derived here. When we plug in our numbers, we find that we actually observe more heterozygotes than what we expect under Hardy-Weinberg equilibrium. And in fact, the excess in heterozygotes is exactly equal to the depletion in the homozygotes. So remember, we had negative 0.0025 for P. We'll have the same thing for Q. Add those two together, and that's the addition that we get into the heterozygotes from the homozygotes. So is this a mechanism of evolution? We've seen that we have violation of Hardy-Weinberg with an excess of heterozygotes and a depletion of homozygotes, but is this actually capable of sustaining change across generations? And the answer is no. One generation of random mating returns the population to Hardy-Weinberg. So let's demonstrate that. So we're just showing here the differences in the frequencies between the sexes. Again, the P is 0.6, P is 0.7, and then that leaves Q for 0.4 of the females and 0.3 for the males. We just multiply those together and write this distribution over here. So our observed capital P, this is the homozygotes, is 0.42, heterozygotes is 0.46, and the other homozygote is 0.12. So from there, we can extract what our new allele frequencies are. So P is equal to 0.42 plus half of the heterozygotes, which is 0.65. And then Q is the same, is equal to 0.35. Now notice that for P, we actually see an increase in the frequency of P for the females from 0.6 to 0.65, and a decrease in the males from 0.7 to 0.65, and we see the opposite for Q, whereas females have lost 0.65, males have gained it. Okay, but now what that has done, since we had this random mating, is that has equalized that frequency between the sexes. And so now we can just represent those frequencies as 0.65 and 0.35 because now they've been equalized. So let's do another generation of random mating. We can see that 0.65 squared is 0.42. This is 2pq here on this diagonal, and then 0.35 squared is 0.12, and we notice that this is exactly what it was in the previous generation. That is to say, we are now in Hardy-Weinberg equilibrium. So one generation of random mating, when there are unequal frequencies between the sexes, returns us to Hardy-Weinberg equilibrium. Therefore, unequal frequencies between the sexes is not a mechanism of evolution. Okay, so we'll just gray that one out. The previous two were mechanisms. This one is not. So now let's look at what the meat of the rest of this presentation is going to be about, and that is the assumption that alleles don't change from the individual to the gene pool. So obviously, when this assumption is violated, that's a mutation, right? This is a mutation being the spontaneous change of an allele from the parent to the offspring. All evolutionary change requires this. Mutations are fundamentally the creative force behind evolution. This is why I started this with this case study showing how a brand new protein emerges from mutational processes. In the absence of any mutational processes, we could not have generated that novel protein. Mutations as a mechanism in evolution have a sort of long and troubled history, and it's likely one of the most misunderstood of all the forces of evolution, and we're going to talk about that in some detail in this video. So the approach is going to be first talking through the classical population genetic approaches to mutation. We're then going to go into the biology of mutations. We'll discuss a little bit about what is meant by the idea that mutations are random. And then we're going to give some examples at the very end of mutations generating novelty. Because again, as I said, mutations are the creative force of evolution. So we really need to evaluate all the different ways in which mutations generate new genetic information. So let's think about mutations as sort of the classical population genetic approach. And this is mutations as an evolutionary force. Let's simplistically say that we start off with biallelic system P and Q, and the frequency of P is equal to 0.5, and therefore the frequency of Q is also equal to 0.5. Let's take the very simple case in which there's only forward mutation in the direction of Q to P. Okay, so, and that occurs at a rate of 0.001. So Q can turn into P, but P never mutates to Q, right? So it's just forward mutation. We can write out the genotypes as P squared equals to 0.25, 2pq is 0.5, and Q squared is 0.25. This is just Hardy-Weinberg proportions. Now, with mutation, we can calculate how the genotype frequencies should change across generations. So, and after one generation of mutation, we should alter the homozygote for P by P plus Pu squared. The reason this is P plus Pu squared is because you have both the first P and the second P, either of which could be added from mutation from Q, okay? And so because of that, we square that value. For the heterozygote, you have two multiplied by P plus PU, because there's some proportion of the heterozygote P that is coming from mutation, multiplied by Q, with a frequency of Q minus some of the Qs that have been lost and converted to Ps. And then the same thing with Q, with you have the frequency of Q minus the number of Qs that are being mutated into Ps. And again, we square it because there are 2 Qs in a homozygote for Q. We then just plug in our numbers and we find that the P squared genotype has increased to 0.2505. The heterozygote has decreased to 0.4999. And again, this is because we're losing some of the Qs to Ps. And then the homozygote for Q is 0.2405. So we can look at how much has it changed. Well, the difference in P is 0.0005. The difference in 2PQ is negative 0.001. And the difference in the Q squared genotype is negative 0.0005. So we can see this, the change in the genotype frequencies are very, very small, even with a relatively high mutation rate, right? So 0.001 is a really high mutation rate, but still it has caused very, very slight changes in the frequency of the genotype after one generation of mutation. We can also look at this at the allele level, which is exactly the same as it is on the homozygote genotype level, just without it being squared. So since the starting frequencies are 0.5 for both, you can write P prime and Q prime as equal to the frequency of P and Q in the next generation. So it's just P plus PU, with PU being the number of P's that are being added from mutations from Q, and that gives us 0.5005. So that's an increase of 0.005 for P, and then obviously Q then decreases by that same amount. Now, as we can see, as I sort of alluded to, this change is very, very slow, right? So we can actually plot this as mutation rate on the x-axis, and then the time for the frequency of Q to change by half on the y-axis. So notice that for even really high rates, like 1e to the negative 5, it will still take 70,000 generations for the frequency of Q to go from 0.5 to 0.25, assuming that the only change that's happening is mutations from Q to P. For a realistic mutation rate of like 1e to the negative 8, it would take 69 million generations to change Q from 0.5 to 0.25. For it to like actually lead to the fixation of P, it would take an inordinately long time. We're talking billions of generations to change Q from 0.5 to 0, right? Now, obviously, this led many early population geneticists to conclude that mutation pressure alone was not an important force in changing allele frequencies in populations, right? Because the rate of change is so small that the pressure that it's applying must be virtually negligible. Now let's also look at the case in which you can have both forward and backward mutations. So there's some rate at which P is going to change to Q, and then Q will change to P. And so in this case, what we're going to do is we're going to replace Q with 1 minus P. Remember, since P plus Q equals 1, then 1 minus P equals Q. And this allows us to just keep up with a single term. So here we have P t plus 1. This is what the frequency of P is going to be after one generation of mutation, is then equal to P sub t 1 minus P. This is the mutation from P to Q, or from P to 1 minus P, plus 1 minus P, which is Q, multiplied by V, with V being the rate of mutation from 1 minus P into P, or from Q back to P. Okay, so we can just simply write it as this, and then from there we can calculate how much change should happen per generation of Q given this sort of back and forth mutational process, as delta P is equal to P sub t plus 1 minus P sub t, where P sub t is what the frequency was in the previous generation, is then equal to V 1 minus P. So again, this is just the rate of change from Q to P minus UP, where UP is the rate of change from P to Q. So then we can ask ourselves, how is this going to impact allele frequencies across generations? So let's look at the change of P when U is greater than V. So that is to say, whenever the rate of change from P to Q exceeds the rate of change from Q to P. And we've just shown a difference in an order of magnitude. So this is where V is 0.0001 multiplied by Q minus 0.001 by P. So this is the rate of change from P to Q, and you can see it's much higher. It's an order of magnitude higher than the rate of change in the opposite direction. I've plotted up here where you can see the rate of change of P is on the Y, and then the frequency of P is on the x-axis. So starting off over here in green, the rate of change goes down until it eventually reaches 0. Now, at a rate of change of 0, this represents an equilibrium point where P is no longer changing, right? So this is where the forward mutation and the backward mutation rates have exactly canceled each other out, and you're at a stable equilibrium. And so when U is greater than V, then that stable equilibrium given these mutation rates is 0.05. So P will persist at a very, very low frequency in the population when the rate of change is greater from P to Q. Now, what about the case in which the rate of changes are exactly equal to each other? So U equals V. We've written that down here in red, so you can see the mutation is 0.001 changing from Q to P, and 0.001 changing from P to Q, right? They're exactly equal to each other. And when that's the case, you can see the rate of change equilibrates or goes to 0 at P equals to 0.5. This is exactly what you would expect, right? Because the same rate of change that's transferring Q to P is transferring P to Q, and so they should persist at exactly equal frequencies with one another. And then lastly is V greater than U, right? So where the rate of change of Q to P is greater than from P to Q, and exactly the same way you can see that the equilibrium is much higher, where P is going to persist at a rate of 0.9. Now we can actually solve for exactly where that equilibrium point should be by simply taking our equation up here, plugging in 0 into delta P, and then solving for P, right? And that's going to give us where that stable equilibrium should be, and we find that it's with P star being the stable equilibrium frequency is equal to V over U plus V. And whenever you plug those numbers in, you can find that you will get exactly these stable equilibrium points. Okay, so that's the classic population genetic approach to mutation on its own, without assuming any other mechanisms, just the mutational mechanism. Like I said, it was considered to be very slow and not a very important process of itself in changing populations. Now, because of this, and because of sort of a long and sordid history that mutation and selection have with one another, we should take a moment and sort of ask the question, what place do mutations have in evolutionary theory? If you've watched my video, People's History of Darwinism, then you are familiar with the Mendelians and the Darwinists and the conflict that they had in the early 1900s. And the Mendelians fundamentally viewed mutation as the central process in evolution. They didn't deny selection per se, it's just that selection was ancillary to the central important mechanism of mutation. Because again, mutation is the creative mechanism of evolution. All variation in populations rely on mutation, and the sorts of variation that you can get is dictated by mutation, right? So that selection to the Mendelians is actually kind of pigeonholed into only operating in certain directions based on what mutation provides to it. Now, this is diametrically opposed to the way the Darwinists and later the architects of the modern synthesis viewed mutation. And in fact, the way I'm going to pitch this is probably the way you've heard it. So they viewed mutation as supplying the raw material of evolution. That is to say that selection sort of picks and chooses from the materials that are supplied by mutation. Now, this basically assumes that mutation is providing the selective mechanism with all possible variation that it can need, that populations are effectively infinite in the amount of variation that they exist with, and so selection has any amount of variation to choose from. And this leads to the idea that mutations are random while selection is purposeful. And the sort of analogy that is often used here is the potter and the clay. That mutations are the clay, and then selection is the potter wielding and shaping complexity out of that clay. And so this, again, led to sort of a setting aside of the mutational process itself as very important in evolution, because, again, it was just assumed that it was constantly providing ample variation for selection to act on. And we sort of rejected the idea that mutation itself was guiding evolution in any particular direction. The history of this is really well reviewed by Arlen Stoltzfus's book, Mutation, Randomness, and Evolution. And he includes in this book a really handy appendix, Appendix C, that has a list of quotations about mutations being random. And so I want to take a moment and read to you some of these quotes, because I think that they will sort of help us to put into context the idea of mutations as they're understood, or as they were understood by the developers of the modern synthesis. All right, so the first quote from Haldane in 1959, he writes, quote, "Variation is in some sense random, but natural selection picks out variation in one direction and not in the other." So from Mayer in 1959, it would be exceedingly difficult to visualize a mechanism by which the environment could induce directly a structural change in DNA molecules that would result in the production of a superiorly adapted phenotype, and more specifically, in the appropriate response to a temporary need. Infinitely variable natural populations are of such evolutionary plasticity that natural selection can mold them in almost any shape. Simpson in 1953 writes, "The ultimate sources mutations are random with respect to selection." Further on, he writes in 53 that the mutations are spontaneous and random, at least in a special sense, elsewhere defined. It is a conclusion warranted and, with some restrictions, demanded by the experimental data. Calling them spontaneous and random means simply that they are not orderly in origin according to the demands of any one of the discarded theories. That the direction of mutations is entirely random is certainly not true, but neither is it true that mutations regularly occur in one direction only. So these are just a couple of the citations from sort of the modern synthetic views about what mutation is and what we mean when we say mutation is random. So where do they get this idea that mutations themselves are not responding directly to the environment, but are instead providing the variation that selection can act upon? Well, a lot of this was established by a very famous experiment by the Letterbergs in 1951. What the Letterbergs sought out to do was to test two competing hypotheses. The first one was pre-adaptation. This is the idea of the modern synthetic thinkers, that in any natural population there exists extensive variation, and so whenever the environment changes, selection then picks from that existing variation, okay? Whereas the alternative hypothesis is directed mutation, that in any population, it is limited by mutational processes, and that when the environment changes, the population responds by these sort of directed mutations that allow them to persist. So not from pre-existing variation, but from a new directed mutational process in response to the environment itself. So they set up a very simple experiment that was quite elegant, and I want to walk you through how it worked. So they started off by plating E. coli bacteria onto an agar plate. And so once those colonies were established, what they did is they then just transferred the colonies from the original plate onto a new plate. And this new plate contained antibiotics. So most of the original colonies died off and only a handful of them survived. Now the question is, did these surviving colonies do so because they were responding to the antibiotics' presence, or did they just happen to possess variation for antibiotic resistance prior to being exposed to it? To figure this out, what they did is they took the original culture and then they just sprinkled antibiotics onto it. Now, if they were responding to antibiotics directly, as opposed to there being some pre-existing variation, it should be random which one of those colonies actually survive, right? But if it was being picked from original variation, then the exact same colonies that existed in the first plating should be the ones that survived after being doused with antibiotics. And that's exactly what the Letterbergs found. They found that the same colonies that survived the initial plating into the new antibiotic medium were the ones that survived back on the original culture, indicating that they possessed some kind of variation for antibiotic resistance before they were ever exposed to it. And so experiments like this is what led many of the modern synthetic thinkers to view mutations as basically, again, providing sort of the raw material for evolution that then natural selection would act upon. So I want to return to this idea, and I want to think about it in light of sort of the modern molecular biological results. But to do this, we need to take a step back and actually talk about the biology of the mutational process itself. Because, again, a lot of the early thinkers didn't really understand like how mutations were being caused and all the different ways in which they're being caused. And unlike the other forces of evolution, mutations are actually like biological things, right? They're happening in the biology of organisms. They're directly connected to their biology, whereas natural selection, for example, is a force of nature, right? It's not something biological. And so I want to like kind of walk us through the biology of mutations. And then what we're going to do is kind of return to this idea, and we're going to reevaluate our conception of the randomness of mutations. What do we mean by that idea? How well is that idea actually held up? And take a sort of more holistic view and see if we can find where mutation should actually fit in evolutionary theory. So to understand the mutational process, we need to understand that most mutations occur during replication. So let's talk through really briefly, just sort of a crash course into how replication happens. So again, what replication is doing is it's unwinding the DNA double helix, and then you're just replicating each of the strands so that you end up with two hopefully identical strands, right? So DNA is bidirectional. You have the 5' to 3' direction, and then the 3' to 5' direction goes in the opposite direction, okay? And so when DNA replication happens, you initially form what's called a replication fork. And this is where DNA helicase comes down, binds to the DNA, and starts to separate it, right? And it starts to separate it by breaking those hydrogen bonds that are holding the nucleotides together. Now, this breaking of these bonds can actually create quite a problem. Imagine, for example, that you have two strings wound together, and then you take the ends of those strings and you try to pull them apart from one another. When you do that, all the upstream strands will start to get tighter and tighter, and will even start to form a kind of supercoil. And the supercoiling can also happen in DNA. So as helicase is breaking that double strands, the supercoils can form upstream. And so what you need then is an additional enzyme that comes and it releases the pressure of that breaking. And that is carried out by the enzyme shown here, topoisomerase. And so again, this prevents that supercoiling from happening upstream. Now, each of the two strands being separated, both of them need to get replicated. One of them is going to get replicated continuously, and the other one is going to get replicated discontinuously. So the one that's replicated continuously is the leading strand, and it is laying new nucleotides from the 5' to 3' direction. That's the direction that DNA polymerase III, which is going to be the one that's actually doing the replication, that's the direction that it can act. And whichever one of those strands that is the continuous process, we call the leading strand. So down here, you can see this is the 5', this is the new one being added, all the new ones are in yellow, and you can see it's being added in the 5' to 3' direction, and then here is DNA polymerase just walking along and adding in the complementary base, okay? So it's following right behind the replication fork continuously. Now, for it to get started, it needs an RNA primer to come and lay down, and that recruits the polymerase and tells it where to begin. Okay, but you only need that one primer. Once polymerase gets started, it's going to replicate continuously. Now, since DNA polymerase can only replicate in the 5' to 3' direction, the lagging strand, that other strand, has a problem. Right, because it needs to be replicated 3 to 5 if it's going to follow the replication fork, but you can't replicate in that direction. So this leads to a sort of discontinuous replication, where as that single strand gets longer and longer, what happens is, again, RNA primers will come and bind that recruits a polymerase that will then walk away from the replication fork. Okay, because it can only do 5' to 3', so it walks away from the replication fork until it reaches a completed double-stranded area, and then it falls off. Okay, and so again, as it's replicating the other direction, more and more of the strand is being broken up behind it, and so you need to then add a new primer and let polymerase start again. And so in this way, you get this discontinuous replication. It also leads to there being little chunks, right? So every time it falls off, there's these little nicks that then need to be repaired along the new strand. And this is fixed by DNA ligase. So DNA ligase comes down, lands on those little nicks, and it repairs them. And each one of those fragments are called Okazaki fragments. So this is the way in which DNA replication is happening. The leading strand is continuously replicated. The lagging strand is discontinuously replicated. Now, importantly, what these polymerases can do is they don't just replicate, but they also have a proofreading mechanism. So as they're walking along the leading strand or the lagging strand in the opposite direction, if they make a mistake, if they put the wrong nucleotide there, they can stop, recognize it, take a step back, and repair it. Okay, so they are capable of doing that. However, if they don't recognize it, they can't go back more than one nucleotide. Once they've sealed the next one, they can't go back and repair 1, 2, or 3 bases back. They have to immediately recognize it to repair it. So that's DNA polymerase 3. So since you have the little RNA primers, you need an additional polymerase that can come and replace them, and that's the job of DNA polymerase 1, which can replace those primers. Now, it also has a proofreading mechanism. It just proofreads in the opposite direction. So instead of in the 3' to 5', it fixes in the 5' to 3' direction. Okay, so that's a really quick and dirty crash course into DNA replication, just so that you can have sort of an idea about this process in your mind as we get into talking about the different actual mechanisms of mutation itself. Okay, so I want to start off by making a very, very clear distinction, is that mutation is not DNA damage. Those two things are not the same thing. Very often when you see people describing mutation, especially if they're like creationist, they will make this equivocation that mutations are the same thing as just damage. That's not what it is. A mutation is the result of inexact repair of damage. So if you have a double-stranded break or a thymine-thymine dimer or any other kind of thing that is DNA damage, and then you have molecules that come and repair them, if they don't repair them exactly as it was before the damage occurred, that's a mutation, right? So it's a very different thing than saying that mutation is damage. It's just inexact repairing of that damage. The first mechanism of mutation I want to talk about is the very simple one that we are all familiar with, and that is the inexact copying and failure to identify the mismatch from DNA polymerase 3 and DNA polymerase 1 that we just talked about. So as I said previously, polymerase 3 is an exonuclease. This means that it can only fix things at the ends of the strand. So it can't go back into the double strand, as I said, and make a repair. It can only do it at the ends. That's the exo component, and the nuclease means it can actually cleave out a nucleotide and replace it with a new one. And polymerase 3, since it has to take a step back, it does all of its repairing in the 3' to 5' direction, opposite the direction it's going to replicate. Polymerase 1, as I said, does the exact same thing. It is also an exonuclease, but it fixes in the 3' to 5' direction. So again, the kinds of mistakes it can make is either a substitution, where it comes in and places an incorrect base. So this is our original strand, CCC, TTT, and then it's Watson-Crick pairing, GGG, AAA. A substitution would be if it replaced a C with an A. Okay, so that would be, initially you would have a mismatch where you had an A and a G. So DNA polymerase can either recognize that mistake, and remember it has to recognize it immediately, can recognize that mistake, throw out the A and replace it with the C, so that then you have a Watson-Crick pair of the C and G. If it doesn't recognize it, then you still have this mismatch, right? You have a C, or you have an A connected to a G, so that's a non-Watson-Crick pair, that still needs to be fixed. So if polymerase doesn't catch it, there's still a sort of backup that can catch it. And this is called base excision repair that relies on DNA glycosylases, which is an endonuclease, to excise the mismatches. And so as opposed to an exonuclease that can only fix on the ends, an endonuclease can actually come into the double strand and excise out nucleotides. So in this case, it would come along, it would recognize that it has an A bound to a G, which is a mismatch, and then it can remove that A and replace it with the correct C. However, a mutation would be if it recognized an A and a G pairing, instead of removing the incorrect A, it removes the G and replaces it with a T. Now, doing that replacement makes it now a Watson-Crick pair, but it's a substitution because that's not what the original base pair was. Okay, so that's one way in which this mutational process can occur. Polymerase can also lead to a deletion where it comes along and misses a nucleotide entirely, or it can accidentally kind of slip and add an extra one. So these are, three different ways in which polymerase, due to the fact that it's not a perfect molecule, it doesn't always put the correct thing, and base excision repair, which is pretty good at coming along and fixing polymerase's mistakes, is itself not perfect and will occasionally make mistakes. And this leads to these sort of single nucleotide polymorphisms. Now, the rate of this in humans is very, very accurately estimated. The germline mutation rate of these sort of single nucleotide variants is 1.17 e to the negative 8. So that's the per generation single nucleotide variant mutation rate. So the second mechanism I want to talk about is one that you've probably heard a little bit less about, and that's error-prone translesion synthesis. And this occurs whenever DNA polymerase 3 stalls out on a damaged site. So imagine, again, where replication is occurring, DNA polymerase is walking along in the 5' to 3' direction, adding in nucleotides. However, if it reaches a site where there's some DNA damage, let's say, for instance, there's a thymine-thymine dimer. This is where you have two thymines that are bound together, right? So this binding together of the two thymines stalls out DNA polymerase. It doesn't know how to keep going because of this damage. Now, this can be pretty bad. If DNA polymerase gets completely stalled out and it can't continue replicating, the entire cell will just die, right? If you can't undergo DNA replication. And so what happens is DNA polymerase 3 will fall off and will get swapped out by two other DNA polymerases, either DNA polymerase 4 or polymerase 5. Now, what these polymerases are capable of doing is actually sort of jumping over the damaged site and continuing to replicate. Right, so it allows replication to continue despite there being a damaged site, and it doesn't result in the death of the cell, because base excision repair can come along after the fact and can repair the DNA damage, assuming you can keep replicating. Now, the key here is that DNA polymerase 4 and 5 are highly error prone. They have very low fidelity, and so they will make a lot of substitutions as they go along because they don't have near the accuracy the DNA polymerase 3 does. And so what you can end up with is substitutions around the site of DNA damage because you've swapped out to a more error-prone DNA polymerase. Now, again, base excision repair can come along and fix some of these, but it may not catch all of them. And so what this ends up generating is a mutational hotspot. So anywhere where you have areas of the DNA that are prone to this sort of damage, especially things like thymine-thymine dimers, you can have mutational hotspots. In yeast, they found that the mutation rate of DNA polymerase 4 and 5 is 100-fold higher than the rate of DNA polymerase 3, and this leads to these mutational hotspots that you've probably heard about. So the third mechanism is nonhomologous injoining, and this is a method for fixing DNA double-stranded breaks. So if you have your DNA molecules, they break, and then there's some level of overhang between the two breaks because they rarely ever just cleave right down the middle. It's normally like broken off in the sides, and so some pieces of DNA are lost. And so in these overhangs, what it will do is it will find a way to rejoin them. Now, that rejoining may not be perfect. So in the figure here, you have CT, which will bind to G and A. And so what it will do is it'll align them and put those two together, but notice that that's not what the original configuration was. So you're actually deleting out several bases all at one time. And this kind of mutation is what's responsible for things like translocations, as well as end-to-end fusions of chromosomes. So you can have really big genes that can get translocated into a completely new location due to this non-homologous injoining mechanism. Another kind is oxidative stress. So in all of our cells, we have mitochondria that is utilizing the electron transport chain to generate ATP. Well, one of the byproducts of the electron transport chain are reactive oxygen species. And these reactive oxygen species are highly damaging the DNA. And they will actually oxidize the bases themselves, and they will cause them to change their conformation, which can be damaging to the cell. And so whenever these bases become oxidized, they need to be repaired. And this repair can either replace it back to what the original base pair was, or it can accidentally replace it with a different base pair. One such example is the oxidized guanine, which is called 8' oxo-DG, and this is often repaired by base excision repair. Now, interestingly, 8-oxo-DG is also an intermediate step in the demethylation of CPG sites, and it serves as a key role in redox regulation. And so I want to talk just a bit about what redox regulation is. So oxidative stress forms these 8-oxo-DG. Again, this is just an oxidized guanine, and this is what it's being represented here. ROS is reactive oxygen species. Right, it's invading the nucleus and it's causing this mutation here to 8-oxo-G. It's oxidizing this guanine. Now, what normally fixes this oxidized guanine is OGG1. This is a protein that will come, recognizes it, binds to it, and will repair it to the original guanine. However, oxidative stress can also deactivate the OGG protein, and so it won't fix it, but it will still bind to that 8-oxo-G site. When it does this, it will actually cause like a 70-degree fold in the DNA molecule itself. And this sharp curve allows for transcription initiation complexes to come and form between these sites. Therefore, it enables gene expression upstream and quite a distance upstream from itself. So this oxidized process is actually quite necessary for the cell if it's going to be able to respond to things like reactive oxygen species or just oxidative stress in general. However, it is error prone, right? So oxidizing guanine still increases the probability that you get a substitution in that site. So despite it being the result of oxidative stress, which is normally pitched as being a bad thing, redox gene expression is an important mechanism in regulating several genes. Another important mechanism of mutation is the deamination at CPG sites. So CPG sites are cytosine, phosphate, guanine site. So it's on one strand, it's just a string of C's and G's. Now this is different than CG content, which is where a C is bound in a Watson-Crick pair to a G, right? So that's CG content. CPG means along one backbone is a C and a G site. Okay, so we add the P in there to show that it's separated by a phosphate between them. Now, these CPG sites are readily methylated by DNA methyltransferases, and I'm showing this right over here. So this is the original cytosine. If you have methylation, then the methyltransferase just attaches this methyl group with this, which is this CH3 right here. And that creates 5-methylcytosine. Now, whenever you methylate one of these groups, it results in the silencing of the genes upstream, right? So this is a process of gene regulation, is methylating these CPG sites. However, these CPG sites have a high rate of mutation due to the process of deamination. So deamination is the loss of this amine group, which is this nitrogen and then H2. So when this becomes lost, that's called deamination. And this results in the generation of a thymine, which is this final thing here. So you have a mutation from a cytosine to a thymine. Now again, mismatch repair can come along, recognize that you have a mismatch between the thymine and the guanine on the other side that it should be paired with. That's a mismatch, and it can either replace the thymine back with the cytosine, or it can replace the guanine with the adenine, right? Repairing it to a Watson-Crick pair. Most of the time, again, mismatch repair is going to, repair it to the correct thing. But since this deamination happens so often, it increases just the number of mutations that you see. And over time, this has led to a depletion in CPG sites relative to what you would expect due to just the GC content in the genome alone. And so what we see in modern genomes is actually what we call CPG islands. So these just islands of CPG sites that are normally associated with genes. So they're very close to promoters of genes because, again, they serve a very important regulatory role. However, most theoretical models show that some ancestral genome would have had lots of CPG sites, but since they are so readily methylated, and since deamination happens so frequently, it has led to the depletion of any CPG site that's not directly connected to gene regulation, right? So that's why we have these CPG islands. Now, this is a pretty big mechanism in shaping genomic content, right? This is not merely a single base thing. This is like the nucleotide content of entire genomes have been shaped by this mutational mechanism. So it's a pretty important mechanism in shaping the way genomes look today. So another important mechanism of mutation is bias gene conversion. And let's walk through how this happens. So this is actually a process that occurs during recombination. So during meiosis, you start off with your two original genomes that then duplicate and they pair together at the centromeres, and then they find one another along the metaphasal plate in meiosis. So when that happens, there's the potential for crossing over, for these homologous chromosomes to actually share content between one another. So let's look at the different combinations that you can get from recombination happening here in this middle area. So the A, B, and C represent different loci. On the blue, they are a capital, and then on the red, they are lowercase. And so you can see you can get all four combinations of these given that recombination is happening here at the middle. So you can get big B, where there's no recombination, or you can get big B, little b, and then little b again with no recombination. And then here on the ends, you can get both C's, little c's, both C's, little c's, all right? So importantly, where the mismatch lies is here in the middle, okay? Big B, little b, little b. Now, what mismatch repair can then do is it's gonna come along and it's going to repair either to the big B or to the little b, because it needs those two to match, right? It recognizes this sort of mismatch right here. Now, presumably, under just equal segregation proportions, the probability that it fixes from big B to little b or little b to big B should be exactly the same. So you should still get an equal proportion in all the gametes. However, if one of those loci preferentially serves as the template for repair, then you get bias gene conversion. So in this case, the big B is serving preferentially as the template anytime you have recombination. So notice that you now have big B's in 3 out of the 4 possible combinations, as opposed to in 2 out of the 4. So they should be half and half. So you get this sort of segregation distortion that occurs due to bias gene conversion. And so this is actually the explanation for many of the GC-rich content that we find in the genome, is where the areas that there are lots of GC content, this is occurring due to bias gene conversion in these recombination hotspots. So it actually allows us to identify recombination hotspots from big chunks that are filled with GC content, because whenever chromosomes are mismatch repaired during recombination, they preferentially repair to the GC-rich areas. So that's bias gene conversion. And again, this is another sort of driving mutational process that can lead to widespread genomic patterns due to this gene conversion. Another really important mutational type is insertional mutagenesis. So this is whenever you get either an endogenous or exogenous piece of DNA gets placed into a new location, normally using the process of reverse transcription. So an endogenous one would be like a transposon, an mRNA transcript, or a mitochondrial transcript. Whereas an exogenous one are something like retroviruses, right? So you get infected with a retrovirus and it inserts itself into your genome. So these are two kinds of insertional mutagenesis that can happen. And in fact, this insertional mutagenesis is one important mechanism of gene duplication. So originally you have a gene region that gets transcribed by mRNA, and that mRNA should then get processed. All the exon or all the introns get spliced out, the exons get pieced together, it should leave the nucleus and get translated. However, occasionally, the pre-processed mRNA can get picked up by reverse transcriptase and inserted someplace else. And this leads to a duplication of that gene. You can also have the same process occurring with a post-processed mRNA. So all the introns have been spliced out, but it gets grabbed by a reverse transcriptase and placed back into the genome. So both of these lead to what are called retrogenes. So these are mRNA transcripts that get accidentally reinserted back into the genome. So this is another important mutational process. Now, this exact mutational process is a key component in what we call adaptive mutagenesis in bacteria via the CRISPR-Cas9 system, which is involved in immunity. This is a really fascinating mutational process, and I want to walk you through how it works. So pictured down here is a bacterial cell, and bacteria, just like us, can get infected with viruses. Viruses that infect bacteria are called bacteriophages. So this is the little bacteriophage, and what it's doing is it's inserting its DNA into the bacteria. Now, what the bacteria can do is a portion of that viral DNA will be recognized by a Cas protein, which lands on it and will cut it. And it will take that DNA and it will insert it into the genome of the bacteria. And this is called spacer acquisition, where what you will have is an array of these viral sequences within what is called a CRISPR array that will then be used as the template for an immune response. So whenever a bacteria will then get reinfected, so another virus will come, land on it, and shoot its DNA into it, what this CRISPR array will do is it'll get replicated, and then each one of these little spacers will become associated with a Cas9 protein. These proteins will then go around to the viral DNA and see if any of them match it. And if one of those RNAs match the viral DNA that was inserted in that spacer, it will bind to it and it will cut it. And that will lead to interference of the infection itself. And so what's really fascinating about this is, one, it's a mutational process, right? It's insertional mutagenesis from the bacteriophage into the bacterial genome. Also, the probability that this occurs increases as the frequency of the bacteriophages in the environment increases, right? So you are more likely to acquire this immunity if there are more bacteriophages there applying the pressure to you. Furthermore, if you can acquire this immunity, you are then more fit against the infection. So there's this interesting thing that emerges here. So it's such that the probability of the mutation increases with the mutation's need. Because again, the need of the mutation should scale linearly with the population size of the bacteriophage. And the probability you get the immunity scales with the population of the bacteriophage. All of these components are linked together. So this is, again, a type of adaptive mutagenesis that the mutational process itself is being driven by the need of the bacteria to respond to infection. So the last one I want to talk about is gene duplication. So we've looked at how insertional mutagenesis can cause this, but there's other ways that this can be caused as well. At a localized level, you can have these small duplications that are caused by polymerase slipping and highly repetitive regions. So like microsatellite regions that are very often used in like court cases, you're identifying close relatives, then you're normally relying on this sort of polymerase slippage in these highly repetitive regions. But large duplications normally occur due to unequal crossing over during recombination. So here's an example where you have this FAB1B protein region, and then you have some regulators before and after it, okay? And so what you expect is that these two, they should align perfectly during meiosis, right? They should be perfectly aligned. But since there are other things that they can also bind to, they may not align perfectly. So the TAT region binds to the TAT region here, right? Because those two things are homologous, so it can bind, but notice it's not binding perfectly. It's just binding on the ends. And what this can lead to is crossing over between those two. If you have crossing over between those two, then that leads to a tandem duplication of this gene region. And so now you have both of the genes. You also now have a shortened chromosome on the other end. So again, this is another mechanism of providing kind of large-scale duplications, is unequal crossing over during recombination. Okay, so now that we've gone through all of these different mutational processes, and there are more, those were just the sort of the highlights that I wanted to touch on. Let's return to this question: are mutations random? And we need to think about what we mean when we say random. What does that actually mean? So first, we could say, well, they're random as in they lack pattern or predictability. This is obviously not true because we can very easily predict where recombination hotspots should be. We can predict the types of mutations that occur at CPG sites. So while they're individually unpredictable, they are more probabilistic as opposed to random in this sort of colloquial usage of lacking any pattern or predictability. They're definitely predictable on sort of a broader scale. So I think this is an incorrect way of thinking about randomness with respect to mutation. Secondly, we could say randomness as individually unpredictable, but that follow a probability distribution, such as rolling a dice. You roll a dice, I can't tell you what the exact roll that you are going to get is, but I can tell you the probability that you roll a 6 or a 5 or a 4. Right? I can tell you the probability distribution of what you will roll. So I think that is a little bit closer to what we mean by randomness and mutation, that they're not individually predicted, but they do follow probabilistic distributions that allow them to be predicted at least probabilistically. Sometimes you hear people say things like randomness with respect to genomic location. This is obviously false. This is like never really been what this idea has meant. Obviously, there are mutational hotspots, as we already said, that are due to error-prone translation repair leads to these mutational hotspots. So we know that those exist. Biased gene conversion due to recombination hotspots. Obviously, randomness with respect to genomic location, there's not a good definition of what we mean by randomness and mutation. So what about randomness with respect to the environment? Right? You don't get certain mutations just because you're in a certain environment. This is also obviously not true. Organisms that live in areas with high UV incidence, for example, have a much higher chance of getting like thymine dimers, for example, which are due to UV radiation. We also saw before in the CRISPR-Cas9 system that bacteria had an increased probability of acquiring a spacer from a bacteriophage if they were in an environment that had lots of viruses. So clearly, mutation are not random with respect to the environment. So what about random with respect to fitness? Now this one's a little bit trickier because mutations, again, unlike the previous mechanisms of evolution, mutations are not independent of fitness. So think about it like this. If a mutation occurs, right, then it is a component part of that organism's fitness. You can't think about this organism's fitness independent of all of its little biological parts. Okay, long fur versus short fur. That is giving an animal a different fitness depending on the environment that it's in, right? Well, long fur versus short fur has a lower level expression that is at the gene level. Right? And those are polymorphisms, those are differences at the genic level. And so just as you can say long fur and short fur are not independent of fitness, right? They are contributing to fitness. The mutations that cause those differences are also contributing to fitness. So you can't divorce fitness from mutation since mutations are a biological part of the organism themselves. So they are not random with respect to fitness in this usage. So the last one that we need to talk about is randomness with respect to their fitness effects. Now this goes back to the Letterberg experiment. That is, do mutations occur because organisms need them as they're entering a new environment, right? Are mutations emerging because of some need? And so their fitness effects and the occurrence of the mutations are not independent of one another. This is what the Letterberg experiment found, and this is generally what the modern synthetic architects meant when they said that mutations are random. They mean random with respect to their adaptive potential. This goes back to mutations are supplying sort of the raw materials of evolution, and so then selection is just kind of picking out from that variation. Now, most of the evidence suggests that standing variation is what is contributing to adaptation whenever a population enters a new environment. Very rarely do we find that it's actually new mutations that are emerging since that population entered the new environment that is then contributing to adaptation. It's almost always standing levels of variation, even if that standing variation is at very, very low frequency. So generally, it's safe to say that mutations don't occur because you need them. right? You're not going to get a beneficial adaptive mutation simply because you need one. But is this an especially useful way of thinking about randomness with respect to mutation? In many respects, and as Arlen Stoltzfus points out in his book, this is akin to saying, I have some crops and I need rain. Right? I need the rain to come and water my crops. But whether or not it's going to rain is independent of my need for that rain. You could say it's random with respect to my need for rain. And while this would strictly be speaking true, it's the weather itself is not random, right? In exactly the same way, mutation itself is not random. It's just random with respect to whether you need it. Right. It's and a better word than random here is independent. It's independent of the need of the organism. It's not occurring because the organism needs it to. Now, of course, as in biology, there are always exceptions. This is like the key thing in biology is that biology is the study of exceptions. And we saw an exception to this exact rule with the CRISPR-Cas9 system, because when the bacteria need the immunity the most is when the viruses in the environment are at the highest frequency. As their frequency increases, the probability of infection increases, which also means that the probability that they acquire a bit of the genome from the virus and therefore a spacer, which will serve as part of the CRISPR-Cas9 system that grants the immunity, also increases. Right. So in this case, the acquisition of the mutation scales directly with the impact of fitness on that mutation. Hence, they are non-random with respect to their fitness effects. So that's one exception to this particular rule. And so in general, we can say that mutations are, and I think we should use the word independent instead of random, that mutations are independent of their effects on fitness. And we can say that mutations are individually random, that is to say, they're individually unpredictable, but they follow a probabilistic distribution. So all of the rest of these things, I think, are incorrect ways of thinking about randomness, and arguably, even the last one here, the word random is not really what we mean. We probably should be saying independent as opposed to random. And again, just kind of thinking about this on a broader scale, how useful is this in thinking about evolution or adaptation? If mutations were not random with respect to fitness, for example, if we with respect to fitness effects, I should say, if we happen to get them more often than not, would that overturn, modern evolutionary theory? Of course not. All that it would mean is that we'd need to incorporate the concept of mutation-directed evolution anytime we wanted to model it, that certain mutations are more likely than others to occur, and then we would incorporate those models into predicting allele frequency changes in populations. And indeed, this is exactly what molecular evolution already does, because we know that some mutations are more likely than others, because we have begun to understand the mutational process itself. And so we can incorporate these mutational biases into our models, and we can see that mutation can filter selection down particular paths, because some mutations are just more likely to occur than others. And so again, the modern synthetic architects were correct in thinking that that mutations are generally independent of their fitness effects, but I think that they were very wrong in thinking that mutations were just the clay, with selection being the potter, whereas really mutations are still kind of guiding the potter in a certain direction, right? They're not merely passive, passengers in the evolutionary epic. They are guiding the story because they're funneling where selection can go, because not all mutations can occur, and they definitely don't all occur with equal probability. So that I think is maybe the key takeaway from sort of the philosophical component of mutations and their role in evolution, now given that we sort of talked about some of the biology of mutations, that we can say they're generally independent of their fitness effects, with exceptions, and that they are individually random, though they follow a probability distribution. So the next time you hear somebody say random mutation, know that there's a lot to this, right? That it's probably more complicated than they think that it is. So the last thing that I want to do, I want to kind of put a little bow on this by talking about the origin of new genes via mutation. I've been trying to build this case that mutations are the creative component of evolution. So let's actually see how they generate novelty. And there's three different ways that I want to talk about. And I'm going to give a specific example for each. And that's de novo gene synthesis, neofunctionalization, and insertional co-option. So de novo gene synthesis is the emergence of a novel gene from intergenic sequences. That is to say, where intergenic regions, that is regions between actual genes that have no function or they have no specific genes in that region, acquire an open reading frame. So an open reading frame is a start codon with some stretch of DNA and then a stop codon. That's an open reading frame, and that is necessary for mRNA to then know where to start, where to stop, and then to take that into a ribosome to be translated. Okay, so you need an open reading frame to acquire a new gene. Now, Durand et al. In 2019 evaluated the emergence of de novo genes from non-coding sequences in Saccharomyces paradoxus. This is actually a species of wild yeast. It's not been domesticated like most of the baker's yeast is Saccharomyces cerevisiae, which is the baker's yeast that is very common as a model system. It's a sister group to this species, and so what they were able to do is kind of look at all the intergenic regions that exist in Saccharomyces cerevisiae and ask, okay, have any of those intergenic regions in Paradoxus acquired genes that don't exist in Cerevisiae? So what they found is that since Paradoxus is a really big population that is spread across the globe, they were able to subset it by populations and then evaluate each of these populations. And indeed, they found that there were de novo genes that were polymorphic within Paradoxus. So some individuals had acquired these open reading frames and some had not. So this little phylogeny here is the relationship between the baker's yeast and then Paradoxus are these three populations here. And then this is a little diagram of the way this might occur. So in black are the genes themselves, and then the purple represents the acquisition of an open reading frame, with ATG being the start codon and TAA being the stop. The ends just represent some codons between them. Now, what Durand found is that for the majority of these open reading frame acquisitions in these intergenic regions unique to Paradoxus, most were very short, and they had low translational efficiency. So they were not translated very often, and so they had very, very low copy numbers, even transcriptionally within each one of the cells. However, around 33 identified de novo genes showed increased translational efficiency that was correlated with the acquisition of an open reading frame. So they actually found that several of these novel genes that arose de novo, so there was no duplication, there was no original gene that they were coming from, they emerged completely de novo from intergenic sequences. 33 of them showed increased translational efficiency. So they were regularly translated in high copy number within the cell. Now, again, this is a polymorphic gene. This is a gene that some individuals of Paradoxus have and others do not have. Right? So it very clearly has emerged since the common ancestor of even just Paradoxus, and definitely since the common ancestor of Paradoxus and Cerevisiae, which are two yeast species. So this is a really fascinating example of how brand new genes can emerge from intergenic sequences. The next one, and the one that's probably a little bit more common, is neofunctionalization. This is where you have an original gene that becomes duplicated, and then this new duplicate copy can acquire mutations that can permit it to obtain a new function. And this new function does not degrade the original function. The original function is still there because it's from a duplication, and so you just have a completely novel function. Now, a really cool example of this is from corn. So maize underwent a recent whole genome duplication event since the ancestor that it shares with sorghum. Both of these are agricultural crops. And so since maize has undergone this whole genome duplication, it not only is just a single gene duplicate, its whole genome got duplicated. Now, what this research found is that around 13% of all the duplicates that exist in the genome have acquired novel regulatory functions. Not only have they acquired novel regulatory functions, but they find that these functions have been maintained by purifying selection since they acquired these functions. They also find extensive evidence not only for novel regulatory functions, but also lots of novel coding functions, and that the maize genome has like 10,000 more genes than sorghum does. And in fact, maize has over 30,000 protein coding genes, which is a pretty large number that are unique from each other, though that clearly share some sort of ancestry because of this genome duplication event. So this demonstrates very clearly that you can have not only novel genes emerge from duplication events, but even novel regulatory pathways evolve because of this duplication. Lastly is insertional co-option. So again, this is akin to insertional mutagenesis, where you have some region that's either being transposed, like a transposon, or some sort of like retrovirus. But the example that I want to give is from retrogenes. So I talked about this before, where these are mRNA transcripts that get reinserted into the genome. Up here on the right, this is a phylogeny looking at retrogene copies across a whole bunch of different organisms. The number of copies are here in this light color. The ones that are in the light blue are retrocopies that are expressed at very low levels. And then the dark blue are ones that are highly expressed retrocopies. And if they have high levels of expression, that means that they are probably functional genes. And indeed, many of these retrogenes have actually co-opted promoters from other regions, including from the CPG sites that we talked about previously, or they have evolved promoter regions de novo. They found that between 88 to 280 retrogenes have been co-opted in each one of the species that they found across the list of vertebrates that they evaluated. What was really fascinating about this is this figure on the bottom right here. So this is human, mouse, platypus, and chicken. And for the human and mouse, they actually have age estimates of these retrogenes. So the light blues are very young retrogenes, and then the dark blues are older retrogenes. The red are then just known protein coding genes. Now, interestingly, the level of expression increases with the age of the retrogene. So those that are particularly old actually approach the level of expression of the protein coding genes themselves, which with young ones being much lower levels of expression. This is exactly what we would expect to see if initially the insertion of a retrogene has no function, right? So it should be expressed at very low levels. It doesn't actually do anything. But over time, as you know, its age increases, there's more opportunities for it to acquire novel function. And as it acquires novel function, including novel regulatory pathways, it then increases its expression as it begins to acquire function. And so that expression level begins to approach the levels of expression that we see in known functional protein coding genes. Okay, so in summary, we've talked about a ton of different things, and so I want to go through the sort of the highlights, the big points that we touched on. First, going back to the very beginning, we found that unequal allele frequencies between the sexes is not an evolutionary mechanism. One generation of random mating returns you back to Hardy-Weinberg equilibrium, and this is why you've never heard of this as a mechanism of evolution. Second is that mutations are the creative force of evolution that generate all novelty. Classical population genetic theory treats mutations as a very weak force in changing allele frequencies, right? We showed mathematically that the forward mutation pressure tends to be very, very weak, and it can take millions, if not billions of generations to change allele frequencies due to just mutations alone, and that in forward and backward mutations, we can actually reach a kind of stable equilibrium. We talked about the biology of mutation, specifically that there are many kinds of mutations, and that they are generally involved in a kind of inexact repair. We talked a bit about the history of mutations and their role in evolutionary theory, that they can be generally described as independent of their fitness effects, and that follow sort of a probabilistic distribution, though are independently random. And we also talked about some exceptions to this, such as the CRISPR-Cas9 system, in which probability of acquiring a mutation scales directly with the benefit that is acquired to that organism via immunity from subsequent viral infection. Lastly, we talked through 3 major sources of new genes and how they can emerge via mutation. We went through an example of a de novo gene synthesis, brand new genes emerging from intergenic regions with no precursors. We talked about neofunctionalization, where you have new genes that are emerged from a precursor, but subsequent mutation change it and allow it to acquire a novel function. And then finally, insertional co-option, where you have an mRNA transcript or even a transposon gets inserted into a new region and then slowly acquires a novel function. So there you have it. The third cause of evolution is mutation. Arguably, mutation is the most important force in evolution, as without it, you would not have evolution at all. Thank you so much for being here. I hope you enjoyed this one. I know, again, this was really information dense. If you have any questions, feel free to drop them in the comments below. If you're interested in this idea about randomness and mutation, I highly recommend you check out Arlen Stoltzfus's book, Mutation, Randomness, and Evolution, in which he dives into the history and background of this debate between sort of the neo-Darwinian or modern synthetic architects and the mutationists that were led initially by the Mendelians and then later by people like Masatoshi Nei that I showed earlier. Again, thanks so much for being here. Hit that like and subscribe, and I will catch you